Deep inside every human cell lies a microscopic landscape of staggering complexity, a spiraling blueprint built from 3 billion chemical letters. This is the human genome, a vast text dictating the physical reality of life. For decades following the 1953 discovery of the double helix structure, this biological code was viewed as an immutable destiny. Scientists could map the precise locations of genes, but the text itself remained locked behind a barrier of microscopic scale and incredible fragility. Navigating this molecular architecture meant attempting to find and alter a single typo buried within thousands of encyclopedias. Early genetic modification attempts were blunt instruments, relying on radiation or viral delivery systems that scattered new genetic material blindly, reaching into the living cell, locating one specific sequence among billions, and cleanly rewriting it was considered a physical impossibility. The biological code was a read-only document, a script humanity could observe but never truly author. The inability to precisely edit this biological text left the medical community largely powerless against hereditary conditions written directly into our DNA. A single misspelled chemical letter out of billions is enough to cause devastating inherited illnesses like cystic fibrosis. Doctors were forced to treat the cascading symptoms of these conditions, managing the physical deterioration of patients while the root cause remained untouchable, locked away inside the nucleus of every cell. Breaking this barrier would mean crossing a fundamental threshold in medical history, shifting the paradigm from managing genetic disease to permanently curing it. The theoretical promise was staggering. If a tool could act as a molecular scalpel, doctors could simply snip out the flawed sequence and paste in the correct one. This dream of precise gene editing fueled billions of dollars in research, though it would eventually spark unprecedented ethical dilemmas. Yet, the breakthrough would not come from the halls of human genetics. Instead of human medicine, the key to rewriting the code of life lay hidden within a microscopic war that has raged across the planet for billions of years. Long before multicellular life evolved, single-celled bacteria were locked in a perpetual struggle for survival against bacteriophages, the predatory viruses that hunt them. These phages constantly inject their own lethal genetic material into bacterial cells to hijack their machinery and replicate until the host bursts. To survive this relentless viral onslaught, bacteria had to develop highly sophisticated, rapidly adaptable defense mechanisms. This ancient microbial battlefield, entirely disconnected from the grand ambitions of human gene therapy, harbored a biological secret of immense power. The solution to humanity's most complex genetic challenges was already perfected by the most primitive organisms on Earth, waiting quietly in the vats of industrial food production. In the late 1980s and early 2000s, researchers studying bacterial cultures began noticing a strange anomaly hidden within microbial DNA. At Osaka University in 1987, scientists sequencing the genome of Escherichia coli stumbled upon bizarre, repeating sequences of genetic code. These were short, palindromic segments of DNA that read the same forward and backward, separated by equally short, seemingly random spacer sequences. At the time, they appeared to be nothing more than biological junk. By the early 2000s, this curiosity shifted into industrial relevance inside the laboratories of Danisco, a Danish food ingredient company. Scientists there were studying the bacteria used to ferment milk into yogurt, trying to understand why their cultures were constantly wiped out by viral phage infections. As they sequenced the genomes of the surviving bacteria, they found those same repeating palindromic sequences, which the scientific community had recently named clustered regularly interspaced short palindromic repeats, or CRISPR. The true function of these CRISPR sequences was a masterclass in biological engineering and adaptive immunity. The mysterious spacer DNA located between the repeating palindromes perfectly matched the genetic sequences of the very viruses that had attacked the bacteria in the past. When a bacterium survived a viral infection, it took a molecular mugshot of the attacker, snipping out a piece of the virus's DNA and physically archiving it within its own CRISPR locus. This archive served as a highly specific inherited memory bank for the bacterial colony. If that same type of virus ever attacked again, the bacterium would transcribe the archived viral DNA into a specialized RNA molecule. This guide RNA would then team up with a CRISPR-associated protein known as Cas, forming a search and destroy complex. The RNA guided the Cas protein directly to the invading virus, where the protein acted as a pair of molecular scissors. While industrial food scientists were mapping this immune response in yogurt cultures, a highly driven French microbiologist named Emmanuelle Charpentier was approaching the puzzle from a vastly different angle. Working in Sweden, Charpentier dedicated her research to Streptococcus pyogenes, the notorious pathogen responsible for deadly, flesh-eating, necrotizing fasciitis. She wanted to understand exactly how this aggressive bacterium regulated its own genes and defended itself against viral threats. Charpentier meticulously analyzed the RNA molecules produced by the flesh-eating bacteria, looking for hidden regulatory mechanisms. The CRISPR system in Streptococcus pyogenes relied on a specific protein called Cas9 to do the actual cutting of viral DNA, but the mechanics of how Cas9 was activated remained incomplete. The known CRISPR RNA, which carried the viral mugshot, was not enough on its own to trigger the Cas9 scissor mechanism. Something else was required to arm the weapon. In 2011, Charpentier published a landmark discovery that finally revealed the missing puzzle piece of the Cas9 system. She identified a previously unknown, highly abundant RNA molecule in the bacteria, which she named transactivating CRISPR RNA, or TRACR RNA. Charpentier proved that this newly discovered TRACR RNA had to link up with the CRISPR guide RNA. Without TRACR RNA, the Cas9 protein remained dormant and harmless, but when the guide RNA and the tricRNA bonded together, they clamped onto the Cas9 protein, activating it and locking it into the correct shape to hunt down and slice through the target DNA. This dual RNA structure was the critical ignition switch for the bacterial immune system. To fully understand its elegant structure, she needed an expert in the physical architecture of RNA. Halfway across the world, at the University of California, Berkeley, Jennifer Doudna operated one of the premier structural biology laboratories on the planet. Doudna had built a world-class reputation by mapping the complex, three-dimensional folded structures of RNA molecules, proving that RNA was a dynamic, active machine capable of catalyzing chemical reactions. Her laboratory was a hub of intense biochemical visualization, capturing the exact atomic shapes of these microscopic engines. Doudna's environment was deeply rooted in the fundamental chemistry of life, driven by a fascination with how the specific folds and loops of an RNA strand dictated its biological function. While she had begun investigating the CRISPR systems of other bacteria, the specific streamlined Cas9 machinery that Charpentier had uncovered was outside her immediate focus. The separate trajectories of these two brilliant scientists were about to collide, setting the stage for a rapid discovery worthy of a Nobel Prize. The intersection of their work occurred in March of 2011 at the American Society for Microbiology conference in San Juan, Puerto Rico. Charpentier, seeking a structural biologist to unravel the atomic mechanics of her TRACR RNA discovery, sought out Doudna. Meeting in a cafe, the two scientists quickly realized the complementary nature of their expertise. Charpentier held the biological key, and Doudna possessed the biochemical tools. Escaping the crowded conference halls, Doudna and Charpentier walked together through the cobblestone streets of Old San Juan, deeply engrossed in a conversation about the machinery of bacterial immunity. By the time they parted ways, they had forged a transatlantic collaboration that would change modern science. They agreed to pool the resources of their respective laboratories, embarking on a joint mission to isolate the Cas9 protein and its dual RNA guides in a test tube detached from living bacteria. The geographical divide between California and the European continent quickly dissolved into a relentless digital collaboration. Following their meeting in Puerto Rico, Doudna and Charpentier synchronized their laboratories, bridging a 9-hour time difference to dissect the molecular components they had isolated. The slow, methodical pace typical of fundamental biological research was suddenly replaced by an urgent, almost frantic exchange of data. The operational rhythm of this joint research effort was sustained by early morning and late night Skype calls, with researchers passing experimental results back and forth across the Atlantic. Postdoctoral researchers Martin Gienek in Berkeley and Krzysztof Kielinski in Vienna became the primary engines of this transatlantic partnership, trading emails filled with biochemical assays and genetic sequences. Their shared goal was to strip the bacterial defense mechanism down to its absolute bare essentials, isolating the machinery in a controlled, artificial environment. To understand the true potential of this system, the teams needed to map the exact structural mechanism of the Cas9 endonuclease. In the wild, Cas9 acts as a pair of molecular scissors, roaming the internal environment of a bacterium to seek out invading viral DNA. But the protein is entirely blind on its own. It requires a navigator to find the specific genetic sequence it is meant to destroy. This navigation system relied on the two separate RNA molecules Charpentier had identified, the CRISPR RNA, which carried the matching genetic code of the virus, and the track RNA, which served as a structural scaffold holding the complex together. When these two RNA strands bound to the Cas9 protein, they initiated a conformational change, locking the molecular machinery into an active state. Once the matching DNA sequence was located, the Cas9 scissors would deploy, slicing through both strands of the viral double helix and neutralizing the threat. By isolating this reaction in a test tube, the researchers arrived at a breathtaking realization that would alter the course of modern genetics. The Cas9 protein was not hardwired to cut only viral DNA, it was completely agnostic to the target. The specific sequence it attacked was determined entirely by the short snippet of CRISPR RNA loaded into its molecular chassis. This meant the system was inherently reprogrammable. If scientists could synthesize an RNA molecule with a sequence matching a specific gene, whether in a plant, a mouse, or a human, they could theoretically direct the Cas9 scissors to that exact location to make a precise cut. The observation of a quirky bacterial immune response had suddenly transformed into the invention of a universally adaptable genetic targeting mechanism. Recognizing the friction of working with a clunky, two-piece RNA navigation system, the researchers decided to engineer a more elegant solution. Gienek and Kielinski began designing experiments to fuse the CRISPR RNA and the TRACAR RNA into a single, continuous molecule. By joining the two strands with a tiny loop of synthetic RNA, they hoped to create a unified guide that could still bind to the Cas9 protein and direct it to a target. The laboratory tests confirmed their hypothesis with stunning clarity. This newly engineered chimera, dubbed the single guide RNA, or sgRNA, functioned perfectly, directing the Cas9 enzyme to slice specific DNA sequences in their test tubes. This crucial engineering step dramatically simplified the system, reducing a complex biological defense mechanism into a streamlined, two-component, user-friendly gene editing tool. The culmination of this frantic collaboration was distilled into a single, densely packed manuscript submitted to the journal Science in June 2012. The academic arena is notoriously rigorous, with peer review often dragging on for months, but the implications of this particular paper were so immediately obvious that it was fast-tracked for publication. The manuscript outlined the complete biochemical mechanism of CRISPR-Cas9 and explicitly stated its potential as a programmable RNA-guided DNA cleavage system. When the paper was published on August 17, 2012, it sent an immediate shockwave through the global scientific community. The pristine, quiet environment of the Berkeley and Vienna laboratories gave way to an explosion of international attention, as the article rapidly became one of the most widely read and intensely debated pieces of biological literature of the decade. The theoretical foundation had been laid bare for the world to scrutinize, replicate, and exploit. For Doudna and Charpentier, the publication brought a profound sense of awe, inextricably linked with a creeping, quiet terror. They understood that by simplifying the molecular scissors, they had effectively democratized the ability to edit the fundamental code of life. The sheer power of what they had created weighed heavily on them, as the boundary between natural evolution and human-directed genetic design began to blur. The realization that this tool could eventually be used to alter human embryos, creating heritable changes that would echo through generations, cast a long shadow over their triumph. The scientific community's immediate recognition of CRISPR's power meant that control over the technology was already slipping from their grasp. The elegant biochemical puzzle they had solved was rapidly mutating into a complex ethical dilemma that no single researcher could contain. The publication of the 2012 paper acted as the definitive starting gun for a new era in molecular biology. The quiet, methodical days of fundamental discovery instantly gave way to a chaotic, intensely competitive rush toward practical application. Researchers who had spent decades struggling with older, more cumbersome gene editing technologies like zinc finger nucleases immediately abandoned their life's work to adopt this new, elegant system. A global scientific race was now underway, fueled by the realization that whoever could successfully adapt CRISPR for use in higher organisms would secure a place in history and potentially unlock billions of dollars in commercial value. Laboratories from Cambridge to Tokyo pivoted their entire operational focus overnight, driven by the urgency to be the first to prove the tool's efficacy beyond the confines of a test tube. The next massive technical hurdle was the eukaryotic challenge. While the single guide RNA and Cas9 worked flawlessly in vitro and inside simple bacteria, human cells presented a vastly different architectural landscape. Eukaryotic cells contain a nucleus, a heavily fortified vault that protects the organism's DNA, wrapped tightly around histone proteins in a complex structure known as chromatin. To make CRISPR function as a medical intervention, scientists had to engineer delivery mechanisms capable of smuggling the Cas9 protein and its guide RNA through the cellular membrane, across the cytoplasm, and into the nucleus. Once inside, the molecular scissors had to navigate the dense, tangled thicket of human chromatin to find a single, specific target among 3 billion base pairs of genetic code, all without making catastrophic cuts in the wrong locations. The definitive breakthrough in crossing this biological frontier did not come from Berkeley or Vienna, but from a fiercely ambitious team in Cambridge, Massachusetts. Feng Zhang, a brilliant young researcher at the Broad Institute of MIT and Harvard, had been independently tracking the CRISPR system and immediately recognized its potential for mammalian editing. Leveraging his deep expertise in eukaryotic systems, Zhang's laboratory successfully optimized the Cas9 machinery to function inside human and mouse cells. In January 2013, just months after the Doudna-Charpentier publication, Zhang's team published their landmark findings in Science, explicitly demonstrating that CRISPR could be used to edit multiple genes simultaneously in living mammalian cells. This monumental success proved that the tool was not just a biochemical curiosity, but a viable technology for human medicine. However, it also set the stage for one of the most bitter and high-stakes intellectual property disputes in the history of science. The pristine, cooperative atmosphere of academic biology vanished almost overnight, replaced by the aggressive, high-stakes theater of corporate litigation. Following the landmark 2013 publications, the United States Patent and Trademark Office became the primary battleground for a multi-billion-dollar conflict over who truly controlled the commercial future of this genetic technology. On one side stood the University of California, Berkeley, representing the foundational discoveries of Jennifer Doudna and Emmanuelle Charpentier, a scientific milestone destined for a Nobel Prize. On the other side stood the Broad Institute of MIT and Harvard, defending the rapid advancements made by Feng Zhang. The air in these legal proceedings was thick with tension, as armies of intellectual property lawyers dissected laboratory notebooks, emails, and publication timestamps to claim ownership over the biological code. The crux of this bitter dispute rested on a highly specific legal distinction between discovering a natural mechanism and adapting it for a novel, non-obvious environment. Berkeley argued that the 2012 paper detailing the single guide RNA was the definitive invention, providing the complete programmable recipe required to operate the CRISPR system anywhere. The Broad Institute countered with the argument that making the system work in complex eukaryotic cells, specifically human and mouse cells, was not a guaranteed or obvious leap from bacterial experiments. They maintained that Zhang's specific modifications, including the addition of nuclear localization signals that allowed the Cas9 protein to breach the human cell nucleus, constituted a separate and legally distinct invention that warranted its own exclusive patent rights. This intellectual property war immediately fractured the scientific community and catalyzed a massive commercial biotech boom. Rather than waiting for the courts to settle the dust, both rival camps rapidly spun out competing, heavily funded companies designed to commercialize their respective claims. Wall Street poured billions of dollars into these newly minted ventures, transforming a humble bacterial defense mechanism into the hottest commodity in modern medicine. Startups like Editas Medicine, co-founded by Zhang, and Intellia Therapeutics, co-founded by Doudna, raced to translate the raw biochemical potential of the molecular scissors into marketable therapies, accelerating the pace of research through sheer financial force. Yet, while the legal teams traded blows over licensing revenues and patent interference claims, the technology itself refused to stall in the courtroom. The true measure of this molecular breakthrough was never going to be found in legal briefs, but in the sterile, high-stakes environment of the clinical ward. The transition from theoretical lab tool to applied clinical therapy happened with unprecedented speed. Researchers pivoted away from institutional rivalries to focus their newly sharpened tools on some of the most devastating and previously untreatable genetic conditions plaguing humanity. One of the most immediate and profound targets was sickle cell disease, a debilitating condition caused by a single-letter typo in the human genome that warps red blood cells into rigid crescent shapes. Rather than attempting to directly rewrite that specific flaw, scientists devised an ingenious workaround utilizing the precision of the editing machinery. The strategy involved aiming the molecular scissors at a specific genetic switch that normally turns off the production of fetal hemoglobin shortly after birth. By cutting and disabling this biological repressor, the gene editing tool forces the body to resume producing the healthy, oxygen-carrying fetal blood cells, effectively compensating for the defective adult hemoglobin. Translating this elegant biological strategy into a living patient involved a grueling, multi-step clinical procedure. Doctors first had to extract millions of blood-forming stem cells directly from the patient's bone marrow, transferring them into a specialized manufacturing facility where the editing could take place safely outside the human body. Inside these sterile cleanrooms, the extracted stem cells were exposed to the Cas9 protein and its custom guide RNA, which executed the precise cuts required to reactivate the fetal hemoglobin. After the patient underwent intensive chemotherapy to clear out their remaining defective marrow, the freshly edited, supercharged cells were infused back into their bloodstream, where they successfully grafted and began producing healthy red blood cells, functionally curing the patients of a lifelong agony. While human medicine captured the global headlines, an equally transformative revolution was quietly unfolding across the world's fields and farms. The agricultural landscape quickly became a massive testing ground for the technology far removed from the sterile confines of the hospital ward. Greenhouses and experimental plots from the American Midwest to rural China began sprouting crops that had their DNA subtly tweaked to withstand the escalating pressures of a changing climate. Researchers rapidly deployed the molecular machinery to engineer drought-resistant wheat, blight-resistant rice, and even livestock capable of surviving devastating viral outbreaks, entirely bypassing the slow, unpredictable process of traditional crossbreeding. This explosion of agricultural and medical innovation was driven by the sudden, radical democratization of gene editing. Unlike previous genetic technologies that required massive institutional infrastructure, the basic components of this new system were astonishingly cheap, easy to synthesize, and simple to use. For less than the cost of a high-end laptop, standard undergraduate laboratories, high school biology classes, and independent biohackers could order custom RNA guides by mail and begin altering the DNA of yeast or bacteria. This widespread accessibility shattered the traditional bottlenecks of biological research, putting the power to rewrite life into the hands of thousands of people who were entirely disconnected from the billion-dollar patent wars. But this same accessibility, combined with the staggering power of the technology, soon cast a long, dark shadow over the initial scientific triumph. The rapid pace of innovation began to outstrip humanity's ability to govern it, shifting the narrative from a celebration of human ingenuity to a profound interrogation of human responsibility and ethics. As the tool proved its efficacy in bacteria, plants, and adult human cells, the inevitable next step loomed on the horizon, bringing with it a gathering storm of complex ethical dilemmas. The scientific community found itself standing at the precipice of a terrifying new frontier, forced to confront the moral implications of an invention that could, if pushed to its limits, permanently alter the trajectory of human evolution. To understand these moral questions, one must grasp a fundamental biological boundary, the difference between somatic and germline cells. When clinicians use CRISPR to treat a patient, they modify somatic cells, like blood or muscle. These edits are localized strictly to that individual. If they later have children, the genetic alterations will not be passed down, making the procedure an advanced form of conventional medicine. Germline editing targets reproductive cells, including sperm, eggs, or early-stage embryos. According to a 2015 consensus report by the National Academies of Sciences, altering these foundational cells means the changes become an indelible part of the organism's entire blueprint. Every subsequent cell division carries the edited sequence, weaving the artificial modification into the resulting adult's reproductive tissues. Crossing this biological threshold carries unimaginable weight. By rewriting the germline, scientists are not merely treating a sick patient, they are making a permanent, heritable alteration to the human gene pool. This grants humanity the power to hijack its own evolutionary trajectory, dictating which traits are passed on and which are erased forever. The permanence of such an act introduces terrifying variables. A single accidental snip in the wrong section of DNA would not just harm one individual, but would be inherited by every descendant that follows. The rapid advancement of the technology meant this was no longer a thought experiment, but an imminent reality demanding answers to deep ethical dilemmas. The sheer magnitude of this responsibility weighed heavily on the technology's pioneers, particularly Jennifer Doudna. As the tool proliferated globally, her initial exhilaration gave way to a creeping dread regarding its potential for misuse. She grappled with the dark historical precedents of eugenics, fearing that well-intentioned medical interventions could easily slip into the pursuit of human perfection. This anxiety culminated in a terrifying nightmare Doudna later detailed in her writings. In the dream, she was introduced to a man who wanted the recipe for her biological breakthrough, only to realize she was face to face with Adolf Hitler. The horrifying imagery encapsulated her deepest fears. Emmanuelle Charpentier shared similar concerns, recognizing that long before any Nobel Prize could validate their work, they had to ensure their creation did not lead to catastrophic harm. Driven by this urgent need for oversight, Doudna and a coalition of leading biologists took decisive action. In December 2015, they convened the first International Summit on Human Gene Editing in Washington, D.C. The summit was a public reckoning, a space where the global scientific community could debate the ethical boundaries of their newfound power. During the proceedings, the organizing committee issued a powerful statement urging a voluntary global moratorium on clinical germline editing. They argued it would be grossly irresponsible to proceed with human pregnancies using modified embryos until safety risks were fully understood. The summit aimed to establish a self-imposed pause, echoing historical conferences that regulated early recombinant DNA research. The summit concluded by drawing a fragile red line. Basic laboratory research on human embryos was deemed permissible, provided it was strictly for scientific understanding and subject to rigorous ethical review. Scientists could use the tool to study early human development in vitro, observing how specific sequences influenced cellular growth. However, an absolute prohibition was placed on implantation. Under the consensus guidelines, no edited human embryo could legally or ethically be transferred into a uterus to establish a pregnancy. This clear demarcation allowed basic science to progress while slamming the door shut on the creation of genetically modified human beings. For nearly 3 years, this self-regulated boundary held, maintaining a tenuous peace within the biological community. Researchers continued to refine the molecular scissors, mapping out new delivery mechanisms and improving precision, all while strictly adhering to the prohibition on human implantation. Then, in November 2018, on the eve of the Second International Summit on Human Gene Editing in Hong Kong, the fragile consensus shattered. A relatively unknown Chinese biophysicist named He Jiankui released a startling video announcement. He claimed to have successfully crossed the ultimate red line, bringing the first gene-edited babies into the world. The revelation exposed a covert rogue clinical trial operating entirely outside international scientific norms. He Jiankui had quietly recruited couples through an HIV advocacy network, seeking out infected fathers and uninfected mothers. Under the guise of a vaccine project, he offered them in vitro fertilization services coupled with an unapproved genetic intervention. Operating in secrecy, his team injected the molecular editing complexes directly into the single-cell IVF embryos of the participating couples. They cultured these altered embryos in the laboratory for several days, sequencing a few cells to verify the DNA cuts before proceeding with the forbidden step of implanting them into the mother's uterus. The biological target of this illicit experiment was a gene known as CCR5. This gene encodes a protein receptor on the surface of white blood cells, which the HIV virus uses as a doorway to infect the immune system. By programming the molecular scissors to disable the CCR5 gene, the rogue scientist intended to mimic a naturally occurring mutation. The theoretical justification was to confer innate genetic resistance to HIV upon the twin girls, protecting them from their father's infection. However, this biological rationale was fundamentally flawed. Standard medical procedures already exist to prevent the transmission of HIV during IVF. Furthermore, disabling the CCR5 receptor increases susceptibility to other dangerous pathogens, introducing entirely new health risks. The announcement triggered an immediate backlash from the global scientific establishment. Leading geneticists, bioethicists, and institutional directors swiftly condemned the experiment as reckless, premature, and profoundly unethical. The international community recoiled at the realization that the technology had been deployed on human subjects before the long-term safety of off-target mutations could be guaranteed. Jennifer Doudna, who had spent years warning of exactly this scenario, expressed deep revulsion at the blatant disregard for the established moratorium. The rogue experiment jeopardized the twin girls' health and threatened to derail legitimate somatic therapies by sparking a regulatory crackdown. The scientific triumph of the decade was suddenly overshadowed by a crisis of human hubris, proving that the ability to rewrite the code of life had vastly outpaced humanity's wisdom to wield it responsibly. The fallout from the rogue experiment transformed theoretical anxieties into harsh legal realities. In December 2019, a Chinese court sentenced He Jiankui to 3 years in prison for illegal medical practices. This unprecedented conviction established a rigid legal boundary, abruptly halting ambitions by other actors to cross the germline threshold. Global health institutions immediately scrambled to construct enforceable guardrails. The World Health Organization convened a multidisciplinary advisory committee to develop standards for governance and oversight. This panel gathered input from scientists and legal experts worldwide to draft comprehensive regulatory frameworks. The resulting 2021 guidelines established a global registry to track human gene editing research and demanded strict whistleblower protections. These sweeping policy mandates cemented an institutional blockade against heritable genetic modification, ensuring future trials would face an impenetrable wall of international scrutiny. As regulatory dust settled, the scientific world sought to reclaim the narrative of discovery. In October 2020, the atmosphere at the Royal Swedish Academy of Sciences in Stockholm crackled with anticipation. The Nobel Prize in Chemistry was awarded jointly to Emmanuelle Charpentier and Jennifer Doudna. The announcement reverberated far beyond Stockholm, validating a decade of relentless laboratory work. For the researchers who mapped the CRISPR-Cas9 protein, the prize offered immense relief. It permanently etched their contributions into the historical record, ensuring the foundational science outshone its early controversies. This victory shattered historical barriers within the highest echelons of recognition. For the first time, a Nobel Science Award was granted exclusively to an all-female team. This milestone fundamentally altered the cultural landscape of molecular biology, serving as an undeniable testament to female leadership. The 2020 Nobel Prize immediately reshaped funding priorities. Doudna and Charpentier's elevation granted them unparalleled authority to steer global science policy, accelerating investments into advanced therapeutics. Their triumph demonstrated that collaborative research could move from basic inquiry to a paradigm-shifting technology in under a decade. Even as the pioneers received their accolades, the technology was evolving beyond the foundational Cas9 system. Researchers engineered iterations like base editing and prime editing, acting more like precise biological pencils. These systems chemically convert one DNA letter directly into another without causing double-strand breaks. These sophisticated iterations drastically reduce off-target mutations. Yet, a critical unresolved question continues to haunt the scientific record. How precisely can we predict the cascading, long-term biological effects of even the most accurate genetic alterations once introduced into a living human ecosystem? This lingering uncertainty bridges the gap between the astonishing pace of laboratory innovation and the heavy burden of clinical application. The journey from studying bacterial defenses to engineering human destiny has permanently altered humanity's relationship with its own biology. The ongoing tension between rapid technological capabilities and collective ethical wisdom remains the defining challenge of the genetic age. As new dilemmas emerge, the ultimate legacy of this biological revolution will be determined not just by molecular tools, but by the moral frameworks enforced across generations. For families carrying inherited conditions, the post-CRISPR reality is no longer an abstract philosophical debate, but a concrete choice presented in quiet clinical consultation rooms. Parents who once faced the certainty of passing down debilitating diseases now look at medical consent forms offering a chance to sever that genetic chain forever. Humanity has permanently crossed the threshold from passive observers of natural selection to active authors of our own biology. The immense weight of holding this molecular pen now rests on every patient, doctor, and regulatory committee tasked with deciding exactly which traits deserve to remain untouched. It all traces back to a microscopic battlefield where a single bacterial cell fought off an invading virus. The elegant, scissor-like action of the Cas9 protein, blindly severing a strand of foreign genetic material, has grown into a power capable of rewriting human destiny. That silent, invisible slice through a double helix remains the defining image of our era, a simple microbial defense mechanism that ultimately handed humanity the keys to its own source code.